The BCL2 family of proteins are critical regulators of intrinsic apoptosis, a highly controlled cell death process. Intrinsic apoptosis is essential for proper development, maintaining homeostasis, and enabling immune responses in metazoans, multicellular animals. This slide provides an evolutionary analysis of how the BCL2 family has been conserved across 700 million years of metazoan evolution, while adapting to diverse functions. The 3D molecular structure of the BCL2 protein family is shown, with the alpha helices highlighted in different colors against a dark background with blue lighting. This detailed scientific visualization helps illustrate the complex and dynamic nature of these crucial biochemical regulators of cell life and death. The BCL2 family of proteins are ancient, with origins tracing back to the earliest metazoans like sponges and cnidarians. These proteins share a common structural feature, the BCL2 fold, which is crucial for their role in regulating apoptosis, or programmed cell death. In addition to the BCL2 proteins, there are also BH3-only proteins that interact with the BCL2 family to modulate their activity and control mitochondrial integrity. The regulation of mitochondrial outer membrane permeability by the BCL2 family is a fundamental function that emerged very early in metazoan evolution, underscoring the importance of this pathway in coordinating cell death processes. Cellular stress, such as DNA damage or pathogen invasion, activates the BCL2 family of proteins. These proteins regulate the mitochondrial outer membrane, causing it to become permeable. This releases apoptosis-initiating factors from the mitochondria into the cytoplasm. These factors then activate a cascade of caspases enzymes that systematically dismantle the cell's components. The dismantled cell is then removed through phagocytosis, preventing an inflammatory response to cellular debris. This intrinsic apoptosis pathway is a tightly regulated process that allows the cell to self-destruct in response to severe internal damage or threats. BCL2 is a family of proteins that play a crucial role in regulating apoptosis, or programmed cell death. These proteins possess up to 4 conserved sequence motifs, called BH domains, BH1-BH4, that form interaction sites and are essential for their function. The BH domains roughly correspond to the alpha helical structure of the BCL2 protein. BCL2 proteins also have a hydrophobic C-terminal transmembrane region that targets them to the mitochondrial outer membrane. This localization allows pro-apoptotic BCL2 proteins like BAX, BAC, and BOC to form pores in the mitochondrial membrane when activated, triggering the apoptotic pathway. Understanding the structural features of BCL2 proteins is key to elucidating how they regulate cell survival and death. The mammalian apoptosis pathway is a complex network of BCL2 family proteins that regulate mitochondrial outer membrane permeabilization. In contrast, the apoptosis pathway in C. elegans is much simpler, with only 2 key BCL2 proteins, CED9 and EGL1. CED9 normally binds and inhibits the caspases initiator CED4 at the mitochondria. When EGL1 binds to CED9, it disrupts this interaction, allowing CED4 to activate the caspases cascade and dismantle the cell. Despite the differences in complexity, the shared outcome of both pathways is the activation of caspases that break down the cell. Today we'll be discussing the research methodology used in this study on BCL2 protein evolution. First, we performed database searches using known BCL2 protein sequences as starting points. This allowed us to identify similar proteins across different species. We then took the sequences we found and used them to iteratively search for more BCL2 analogs, building up our dataset. Next, we constructed phylogenetic trees to understand the evolutionary relationships between the BCL2 proteins we identified. This involved using maximum likelihood methods and bootstrap analysis. We also validated the gene structure and synteny of the BCL2 proteins by cross-referencing the NCBI database. Finally, we analyzed the structural features of the BCL2 proteins, looking for the characteristic BCL2 fold and BH motifs. We used tools like the Conserved Domain Database and DALI structural alignment to identify these key structural signatures. The structures of BCL2 family proteins from basal metazoans are remarkably similar, despite having relatively low shared sequence identities. This structural alignment reveals the importance of the conserved backbone structure for the function of these proteins. The hydrophobic core and the BH3 binding groove formed by helices alpha-2, alpha-5 and alpha-8 are critical features shared across the BCL2 family. The conserved residues in the BH1-BH3 motifs provide a binding surface that is essential for the interactions of these proteins. Evolutionary analysis based on the structures shows that Hydra and Trichoplax BCL2 structures are closely related, while the G-Sidonium BHP2 structure clusters with those from bilaterians like zebrafish and human BCL2. This highlights the structural conservation of BCL2 proteins across diverse metazoan lineages, suggesting their fundamental importance in cellular processes. The slide presents data on the number of BCL2 genes found in the genomes of several basal metazoan species. BCL2 genes play a key role in regulating apoptosis, or programmed cell death, in multicellular organisms. The sponge Amphimedon queenslandica has 7 BCL2 genes, the Cnidarian Hydra vulgaris has 11, and the placozoan Trichoplax adhirans has 4. In contrast, the Stenophore Nemeopsis leadii appears to have lost the BCL2 family and related apoptosis genes entirely. The presence of multiple BCL2 paralogs in each species indicates that complex BCL2 signaling was established early in metazoan evolution. This suggests that apoptosis regulation was an important feature even in the most ancient multicellular animals. The BCL2 family of proteins play a critical role in regulating apoptosis, or programmed cell death. There are 3 main groups within the BCL2 family: pro-survival proteins, pro-apoptotic proteins, and BH3-only proteins. The pro-survival proteins like BCL2, BCLXL, and MCL1 inhibit apoptosis by binding and sequestering the pro-apoptotic proteins. The pro-apoptotic proteins like BAX and BAC can form pores in the mitochondrial membrane, leading to the release of cytochrome C and triggering cell death. The BH3-only proteins like BIM and PUMA can neutralize the pro-survival proteins or directly activate the pro-apoptotic proteins, tipping the balance towards apoptosis. There is also a group of unclassified BCL2 proteins from basal metazoans that don't readily cluster with the pro-survival or pro-apoptotic groups. The key pro-survival BCL2 proteins in mice generally have a 3-exon structure, with the first exon being untranslated. The coding region is formed by splicing exons 2 and 3, with the C-terminal transmembrane region on exon 3 separate from exon 2, which contains the BH3, BH1, and BH2 motifs. In contrast, the pro-apoptotic BCL2 proteins like BAX, BAC, and BOC have a more complex gene structure with 4-6 exons. An interesting difference is that the BH3 motif in BOC is split between 2 exons, unlike the other BCL2 proteins. The exon 2, exon 3 boundary is highly conserved, with exon 2 ending at a conserved tryptophan in the BH2 motif, GGW. This conservation extends to basal metazoans like Amphimedon queenslandica and Trichoplax adhirans, whose BCL2 proteins show similar exon boundaries. The image shows an evolutionary tree of bilaterian animals, with the branches colored to represent the distribution of BCL2 genes across different groups. Ecdysozoans, like the nematode C. elegans, have reduced numbers of BCL2 family genes, with only a single BCL2 protein, while flies have 2 BCL2 genes. Lophotrochozoans, on the other hand, have multiple BCL2-like genes and a more complex apoptosis signaling system comparable to mammals. The deuterostomes, which include vertebrates, show a complex pattern, with some members undergoing extensive gene loss, while others maintain multiple BCL2 proteins. Vertebrates have an expanded number of BCL2 family members due to gene duplication events, with mechanisms similar to those found in mammals. Stenophores, sea jellies, the tunicate Oikopleura dioica, the aquoil flatworm Hofstenia myanea, and certain nematodes like Meludogyne species have lost all BCL2 family genes. This indicates that BCL2-regulated apoptosis is not an absolute requirement in metazoans. Despite lacking BCL2 genes, many of these organisms still possess caspases genes, suggesting they may utilize alternative pathways for programmed cell death. Recent gene deletion studies in mice have also shown that the BAX, and BOC pathway is not strictly necessary for developmental apoptosis. This curious case of gene loss challenges the assumption that BCL2-regulated apoptosis is essential across metazoans. In terms of BH3-only proteins, later evolutionary arrivals. It shows the evolutionary timeline of BH3-only proteins, which are an important class of proteins involved in programmed cell death. Starting with the earliest life forms like Porifera and Placozoa, which have no identifiable BH3-only proteins. Moving to Cnidarians, where we see the first appearance of BH3-only proteins. Then Echinoderms, which have potential BH3-only proteins. In chondrichthyes, cartilaginous fish, the key BH3-only proteins BIM, BMF and BID appear. Finally, in mammals we see the full complement of BH3-only proteins. This slide provides a high-level overview of how these important cell death regulators evolved over time. The BH3 motif is the only conserved sequence element in these otherwise diverse BH3-only proteins. Average intrinsic disorder prediction, IDP, indices greater than 0.5 indicate that potential BH3-only proteins in cnidarians and echinoderms have largely unstructured regions. The sequence length of potential echinoderm BH3-only proteins ranges from 118 to 373 amino acids. This intrinsically disordered nature allows BH3-only proteins to function as hub proteins that can interact with multiple partners and undergo conformational changes when binding to their pro-survival BCL2 targets. The occurrence of BIM, BAD, and BMF prior to BID suggests that the intrinsic disorder of BH3-only proteins is fundamental to their pro-apoptotic action. APAF1 is a critical component of the intrinsic apoptosis pathway, acting as an apical caspases activator to initiate cell dismantling. Caspases are the executioner proteases that carry out the actual destruction of cellular components during apoptosis. They are present even in organisms that have lost the BCL2 genes. Cytochrome C is normally sequestered in the mitochondria, but is released during MOMP, mitochondrial outer membrane permeabilization, to activate APAF1. However, in some species like starfish, cytochrome C may not be necessary for APAF1-mediated apoptosis. MOMP is a key regulatory step in intrinsic apoptosis, and the proteins that control it can vary significantly across evolutionary lineages. Apoptosis, or programmed cell death, is a fundamental biological process that is highly conserved across metazoan lineages. This slide provides a comparative overview of key apoptotic mechanisms and their evolutionary conservation. We can see that core components like BCL2 proteins, BH3-only proteins, APAF1, and cytochrome C release are present to varying degrees across different organism groups. Some interesting patterns emerge, like the apparent absence of these pathways in stenophores, and the reduced or variable nature of apoptotic mechanisms in ecdysozoans like C. elegans. The diversity and conservation of these pathways underscores the critical role of apoptosis in metazoan biology and development. Understanding the evolutionary history of apoptosis can provide valuable insights into the fundamental mechanisms of cell death and their importance across the tree of life. Early metazoans like placozoans, hydra, and echinoderms provide insights into the evolutionary origins of apoptosis. Placozoan BCL2 homologs localize to mitochondria and can induce apoptosis, suggesting mitochondrial apoptosis is possible in these simple animals. Hydra BCL2 proteins interact and function similarly to their mammalian counterparts, indicating evolutionary conservation of this pathway. Echinoderms show variation in how the apoptotic pathway is triggered, with cytochrome C playing different roles in different species. These studies on early metazoans help us understand the ancient origins and diversification of the apoptotic machinery. C. elegans has a single BCL2 protein called CED9 that binds and inhibits the caspases initiator CED4 at the mitochondrial outer membrane. The BH3-only protein EGL1 antagonizes the CED9, CED4 interaction, allowing CED4 to be released and activate the caspases cascade. The activated caspases then dismantle cellular components without mitochondrial outer membrane permeabilization, MOMP. This direct scheme of caspases activation is not mirrored in mammals, where a complex network of interactions regulates MOMP. Some nematodes in clade IV have lost all BCL2 family genes, indicating alternative mechanisms for programmed cell death in these organisms. BH3-only proteins are a class of BCL2 proteins that are predicted to be intrinsically disordered based on computational analysis. The graph shows the intrinsic disorder prediction scores for 3 BH3-only proteins: human BIM, human BAD, and a Hydra BH3-only protein. Scores above 0.5 indicate unstructured, disordered regions, which is a key feature of BH3-only proteins. The data suggests this intrinsic disorder is conserved across evolutionary lineages, from humans to the Cni-Darien Hydra. This disordered nature is important for the function of BH3-only proteins in regulating apoptosis and other cellular processes. The evolutionary timeline of the BCL2 family of proteins provides insights into the development of apoptosis regulation across different organisms. We can see that BCL2 proteins first appear in early metazoans like sponges, placozoans, and cnidarians, but are absent in their sister groups like choanozoans and philisterians. The addition of BH3-only proteins in cnidarians added further complexity to the regulation of apoptosis. Bilaterian organisms show diverse patterns of BCL2 family gene conservation, expansion, and loss across different lineages. Vertebrate evolution, particularly in teleost fish, involved gene duplication events that expanded the number of BCL2 family members. In mammals, we see a complex network of 17 BCL2 family members that regulate the intrinsic apoptosis pathway. The key structural feature of BCL2 proteins is the BH3 in-groove mechanism, where conserved residues in the BH3 motif of a ligand bind to a groove formed by the BH1-BH3 motifs on the receptor BCL2 protein. This BH3 in-groove binding mechanism has been evolutionarily conserved from the earliest metazoans, like sponges, all the way up to humans, despite relatively low sequence conservation in BCL2 proteins. The conservation of this fundamental interaction mechanism across 700 million years of evolutionary history suggests that the basic regulatory principles of apoptosis were established early on and have been maintained in metazoans. The BCL2 gene family has a highly conserved structure across evolutionary time, from sponges to mammals. The pro-survival BCL2 genes have a 3-exon structure, with the first exon being untranslated and the coding region formed by exons 2-3. The exon 2, exon 3 boundary is located at a conserved tryptophan in the BH2 motif, which is a key functional domain. The BH3, BH1, and BH2 motifs that are critical for BCL2 protein function, all located on exon 2, while the transmembrane region is on exon 3. This conserved gene structure and domain organization has been maintained since the earliest multicellular organisms, demonstrating the evolutionary stability and importance of the BCL2 family. The mitochondria are crucial organelles that play a key role in cell function and survival. BCL2 proteins, which regulate apoptosis, program cell death, are localized to the mitochondrial outer membrane. This localization is enabled by a hydrophobic C-terminal transmembrane region in the BCL2 proteins. In addition to the mitochondria, some BCL2 proteins like BOC are also found in the endoplasmic reticulum and Golgi membranes. Pro-apoptotic BCL2 proteins like BAX, BAC, and BOC can oligomerize and form pores in the mitochondrial outer membrane. This mitochondrial outer membrane permeabilization, MOMP, leads to the release of apoptosis-initiating factors, triggering the caspases cascade and cell death. Understanding the localization and function of BCL2 proteins is crucial for studying the regulation of apoptosis in cells. Cancer is a disease characterized by the dysregulation of BCL2 family proteins, which play a critical role in regulating apoptosis or programmed cell death. Understanding the evolutionary conservation of these proteins helps identify their most fundamental functional regions, which can then be targeted by drugs like BH3 mimetics. Neurodegenerative disorders like Alzheimer's and Parkinson's disease also involve inappropriate activation of apoptosis, leading to neuronal loss. Evolutionary insights into apoptotic regulation can reveal which aspects are most essential. Many viruses have evolved to target the BCL2 pathways in order to prevent host cell death and promote their own replication. The evolutionary arms race between hosts and pathogens has shaped the diversity of BCL2 family proteins. Evolutionary analysis of BCL2 proteins can also help predict potential off-target effects and resistance mechanisms for therapeutic BH3 mimetic drugs, informing their further development. The BCL2 family of proteins play important roles beyond just regulating apoptosis or programmed cell death. These proteins also have non-apoptotic functions in calcium homeostasis, mitochondrial dynamics, and autophagy regulation. BCL2 proteins can modulate calcium release from the endoplasmic reticulum, impacting cellular signaling pathways independent of apoptosis. Some BCL2 proteins may lack the ability to bind BH3-only antagonists, suggesting they have specialized non-apoptotic functions. BCL2 proteins can influence mitochondrial fission and fusion, affecting cellular metabolism and energy production. This may represent an ancient role that predates the evolution of regulated cell death pathways. Several BCL2 family members interact with autophagy machinery, providing a link between different cellular quality control mechanisms. This connection may explain why some organisms can survive the loss of BCL2 genes. Overall, the non-apoptotic functions of BCL2 proteins highlight their diverse and critical roles in cellular homeostasis and physiology. The slide discusses the independent evolution of apoptotic pathways across different metazoan lineages. Sequence searches show that the BH3-only proteins in schistosomes and nematodes are not phylogenetically related, suggesting they arose through convergent evolution. The absence of BCL2 genes in some metazoans indicates that mitochondrial-activated apoptosis is not an obligate feature, and these organisms must use alternative mechanisms for programmed cell death. Even in lineages that retain BCL2 proteins, there are differences in apoptotic mechanisms. For example, Hydra have a mammal-like tripartite apoptosis signaling, while placozoans may use a BAC-like protein instead of BH3-only proteins. The slide includes a diagram illustrating the convergent evolution of apoptotic pathways across different metazoan lineages. Hydra vulgaris, a cnidarian, has an expanded BCL2 family with many members. C. elegans, a nematode, has a single BCL2 protein, Mus musculus. The mouse also has multiple BCL2 family members. Oikopleura dioica, a tunicate, has BCL2 family members. Many early metazoans have a greater number of BCL2 genes compared to later metazoans. This contraction in BCL2 genes corresponds with the development of adaptive immune responses, suggesting a potential trade-off between different cellular defense mechanisms. Teleost fish like Danio ririo have experienced gene duplication, leading to an expanded BCL2 family. In contrast, ecdysozoans have lost or eliminated most of their BCL2 proteins. Bok proteins are a relatively understudied pro-apoptotic member of the BCL2 family. Yet they have some unique features that set them apart from other BCL2 proteins. Bok proteins are present in species as early as cnidarians, one of the most primitive multicellular organisms. The BH3 motif of Bok proteins spans 2 exons in mammals, unlike other BCL2 proteins. Unlike other pro-apoptotic BCL2 proteins, Bok localizes to the and Golgi membranes, not just the mitochondria. These specialized features of Bok have been evolutionarily conserved across diverse lineages, suggesting an important and ancient function. Cytochrome C is a small protein found in the mitochondria that plays a key role in the electron transport chain. During apoptosis, or programmed cell death, cytochrome C is released from the mitochondria into the cytoplasm. This release of cytochrome C serves as an important signal that triggers the activation of caspases, which are enzymes that carry out the cell death process. In mammals, cytochrome C binds to a protein called APAF1, causing it to oligomerize into a structure called the AP optosome. The AP optosome then activates initiator caspases, which set off a cascade of caspases activation leading to apoptosis. Interestingly, the requirement for cytochrome C in apoptosis varies across different species. In the starfish Asterina pectinifera, cytochrome C does not appear to be necessary for APAF1-mediated apoptosis. The BCL2 family of proteins appears early in metazoan evolution, but is absent in some lineages, indicating it's not an absolute requirement for multicellularity. Despite sequence divergence over evolutionary history, the 3D structure of BCL2 proteins has remained essentially topologically invariant. Lineage-specific diversification and gene losses have contributed to numerous differences in how different organisms utilize these apoptotic proteins. Understanding the evolutionary relationships and variations in BCL2 proteins may provide insights into their roles in normal function and disease. Future research should focus on characterizing unclassified BCL2 proteins, understanding alternative apoptotic mechanisms in organisms that have lost BCL2 genes, and exploring the non-apoptotic functions of this ancient protein family. These evolutionary insights could lead to new therapeutic approaches for diseases involving dysregulated apoptosis.